This is everything you need to know for Unit 5 of AP Biology. We're going to go ahead and start off with chromosomes. This is a pretty dense unit, so we'll try to move fast and cover everything quickly. So what do you have to know about chromosomes? In humans, our somatic cells have 46 chromosomes, and each chromosome is a single-stranded DNA molecule that's combined with proteins. 23 of those molecules come from your mom, and 23 of those come from your dad. So cells, remember, are either in interphase or M phase. When they're in interphase, they're in either G1, S, or G2. And S is when the DNA in the cell is duplicated. So if you look at a cell that's in G1 of interphase prior to S phase, this is what it'll look like. You'll have 46 chromosomes. Each chromosome is a single DNA molecule. There are essentially 46 lines or 46 chromosomes. The cell is diploid. 2n equals 46. Those 46 chromosomes come in 23 pairs. And each pair is of a particular shape and size. So out of those 23 pairs, 22 pairs are autosomes and 1 pair is a sex pair. And that sex pair is either XX or XY. So what does the cell look like after S phase of interphase? So you still have 46 chromosomes, but this time the chromosomes are not single molecules like one straight line. You don't have 46 straight lines anymore. You now have 46 X's because each of those chromosomes that were previously just one single DNA molecule have duplicated. And now they look like an X with 2 sister chromatids connected at the centromere. But you still have 46 of these X's, and 23 of them will have come from mom, and 23 of them would have come from dad. So that's what you have to know about chromosomes in terms of their numbers and what they look like in a cell. So let's go ahead and move on to the second topic, meiosis. So meiosis is split into meiosis 1 and meiosis 2. In meiosis 1, you have homologous chromosomes separating. That means from each pair, one pair, which looks like an X, goes to one daughter cell, while the other pair, the other X, goes to the other daughter cell. So out of the 46 total chromosomes, you have essentially a lineup of 23 next to each other. And then 23 go to one, 23 go to the other. Okay, meiosis 2. Sister chromatids separate in meiosis 2. So each daughter cell has 23 chromosomes, but each chromosome has 2 sister chromatids, right? So in meiosis 2, those 2 sister chromatids separate, and you end up with 4 daughter cells this time, and each daughter cell has 23 chromosomes. They're haploid, and each chromosome now is a single DNA molecule. It's no longer an X, and that's the end of meiosis 2. So let's dig deeper into meiosis 1. So G1 has passed, S has passed, G2 has passed, and now you're in prophase 1 of meiosis. So in prophase 1, what happens? Chromosomes condense. The sister chromatids from homologous chromosomes cross over at sites called chiasmata. The centromeres, the centrosomes, they start moving apart and spindles start to form, and then the nuclear envelope disintegrates. That's prophase 1. Next, metaphase 1. Metaphase is always simple to remember. That's when all the chromosomes will align at the metaphase plate in the middle. But what you have to remember is what is aligning. So for metaphase 1, you have homologous chromosome pairs aligning at the metaphase plate. So that means down the plate, you'll have 2 X's right next to each other. One X will have been from one parent, while the other X will have been from the other parent. So how many of these X pair-ups will you have down the metaphase plate? You'll have 23 of them. And then in anaphase 1, those homologous chromosomes, those X's, separate. So one daughter cell will get 23 X's, and the other daughter cell will get the other 23 X's. That's anaphase 1. And then telophase 1 and cytokinesis is simple enough. That's when the cytoplasm will split, and you'll have essentially 2 daughter cells. Each have 23 chromosomes, but remember, each chromosome is a X. It appears as X because it's essentially 2 DNA molecules bound at a centromere. What about meiosis 2? In meiosis 2, it begins with prophase 2. The nuclear envelope may form and deform again. You might have, again, some spindle formation, but that's kind of not all that important. What you have to focus on is more so metaphase 2. So metaphase 2, this time you have chromosomes, individual chromosomes that appear as X's lining up at the metaphase plate, and then the spindles attach, and then in anaphase 2, those sister chromatids separate. And they separate, and you end up with now 4 daughter cells in telophase 2/cytokinesis of meiosis 2. You have 4 daughter cells. They're all haploid. They have 23 chromosomes, and each chromosome is now a single DNA molecule. And that's all you need to know for meiosis 1, meiosis 2 of meiosis. Okay, let's move on to the third topic, which is Punnet squares. We're going to go through 3 hallmark Punnet squares and the ratios that you need to know, and it just needs to click off from the top of your head whenever you see these ratios. So the first is the 3:1 Punnet square ratio. So what do you have to know about Punnet squares? Number 1, a character is a phenotype, like, say, flower color. A trait is a variant of a phenotype, so white flower color and purple flower color. Those are traits. True breeds are plants that produce the same phenotype across multiple generations because they have the same homogeneous gene composition. They don't have different alleles. We're going to talk about alleles in a second. So in Mendel's experiments, he was using pea plants, and pea plants have a carpel, which is like the female egg, and then they have a stamen that produces pollen, which is like the male sperm. So you have to know about those 2 components. So typically that single pea plant has its own stamen, its own egg, and its own carpel. Its own stamen, which produces pollen, its own carpel, and it self-fertilizes. But Mendel, what he would do is he would cut off the stamen, which is the sperm-producing part, and he would take pollen from another plant and then brush it onto the carpel of the other plant. And he would basically cross plants in this way and control it in that way, and that's what we call crossing a plant or hybridization. So you can visualize hybridizations in a Punnet square. A Punnet square lets you predict the allele composition of offspring when the genotype of the parent is known. So our character that we'll look at right now is flower color. The traits will be purple and white, and the dominant allele, capital P, encodes purple, while the recessive allele, little p, encodes white. The flower color gene, we'll assume right now, is on an autosome. So what happens when you cross a homozygous dominant purple flower, double P, capital P, with a homozygous recessive white flower, little p? So the F1 generation, they're all big P, little p. They're heterozygous, right? And that's because only one parent can only donate a big P allele, and the other parent can only donate a little p allele. So the F1 generation is pretty simple. The F2 generation is more interesting because each F1 parent can now donate either a big P and a little p, or a big P and or a little p. Do you get that? So in the F2 generation, it's more interesting because each parent can either donate a big P or a little p. And the cross ends up looking like this. A quarter of the time, the child will be big P, homozygous dominant. A quarter of the time, the child would be little p, homozygous recessive. And half the time, the child will be heterozygous, big P, little p. That's genotypic makeup. The phenotypic makeup is 3/4 purple and 1/4 white, hence the 3:1 ratio. 3:1 ratio, you should recognize immediately, and you should know what's going on. So the second topic is the Punnet squares with a 1-to-1 ratio. This is what we call a test cross. So the second Punnet square you should know is a test cross. It's where you have a flower of an unknown genotype that's crossed with a flower of a known homozygous recessive genotype. So for example, a purple flower with an unknown genotype, it might be big P, or big P, little p. It might be homozygous dominant or heterozygous, we don't know. It's crossed with a white flower with a known genotype, little p, Crossing that unknown purple flower with a known homozygous recessive flower helps flesh out what kind of purple flower it is. It could be homozygous dominant, or it might be heterozygous or heterozygous. So let's work out through the two test crosses. So in the first case, if the purple flower is homozygous dominant, then you can only donate a big P. In that case, all the children will be big P, little p. But in the second case, if that purple flower is big P, little p, aka heterozygous, then it can donate either of the two. And what you end up getting is half the children will be big P, little p, and half the children will be little p, little p. That's the genotypic makeup, while the phenotypic makeup is half purple, half white. So that's a 1-to-1 ratio. Third and final Punnet square, we're going to actually go through 4. The third is the 9-3-3-1. So this is when we're moving into dihybrid crosses. So let's look at, to stay consistent probably with your textbook, we'll stick with seed shape and color. So everything you've been learning so far is talking about the law of segregation. That's where alleles are donated separately into each gamete. And now we're going to talk about independent assortment. So let's look at P color and P shape. So for P color, capital Y is yellow dominant, while little y is green recessive. And for P shape, big R is round and dominant, while little r is wrinkled and recessive. So let's cross a double homozygous dominant with a double homozygous recessive. Okay, so that's a big Y, big Y, big R, with a little y, little y, little r, little r. Remember that during gamete formation, each gamete needs to have an allele for each gene. So that gamete needs to have an allele for the P color and it needs to have an allele for the P shape. So the first parent, it can only donate a big Y and a big R for shape and color. The other parent, that can only donate a little y and a little r for shape and color. So that means after fertilization, the F1 offspring will all be big Y, big Y, little y, big R, little r. That's phenotypically yellow and round, but they're all heterozygous. Okay, now the F2 offspring is more interesting. This time, each parent can donate either a big Y, little y, or a big R or little r. So this is how you kind of divvy it up in your brain. What you do is, okay, if a parent donates a big Y gamete, it can associate with a big R or a little r. So you get a big Y, big R, and big Y, little r gametes. Now, if a parent donates a little y gamete, it can pair with either a big R or little r again. So you have a little y big R and a little y little r. So those are the 4 different types of gametes that each parent can make. And now if you line them up on a Punnet square at the top and along the top and along the side, and you do out the math, what you get is out of 16 of the progeny, 16 because you add the 9, the 3, and the 1, and you get 16. Out of those 16, 9 will be yellow and round, 3 will be yellow and wrinkled, 3 will be green and round, and 1 will be green and wrinkled, like having both the recessive traits. So 9 will have both the dominant phenotypes, and 1 will have both recessive phenotypes, and then those middle 3 threes will be 1 dominant, 1 recessive, 1 recessive, 1 dominant. And this is essentially demonstrating the law of independent assortment because alleles for seed color and seed shape, they're assorting independently. All right, and so the last Punnet square that you have to know is a 9-3-4, and that comes from a phenomenon called epistasis. So epistasis is when the expression of one gene affects the expression or phenotype of another gene. So let's look at coat colors in labs. Black, big B is dominant, while brown is little b recessive. There's a second gene that determines pigment deposition, and successful deposition is big E and dominant, while the failure to deposit pigment is little e and recessive. When a pigment fails to deposit, the lab ends up having a yellow coat. And this overrides the presence of having a big B or a little b. It doesn't matter, you don't get black or brown, you get yellow when you have little e, the little e recessive allele, two of them, okay, because it's recessive. So what happens if you cross two black labs that are heterozygous for both the coat color and the deposition gene? So big B, big E, little e, if you cross two of those. So out of the 16, 4 of them will get little e alleles, and they won't be able to deposit pigment, and they'll be yellow. So that's 4. And then if you do the math and you just do the, put in the gametes in the top and the side, and then cross them and count them up, 9 will be black, of like varying genotypes. So it'll be big B, or big B, little b. And then 3 will be brown, little b. And then 4 will have the little e, won't be able to deposit pigment, and will be yellow. So that's where you get your 9:3:4 ratio from epistasis. Okay, let's move on to incomplete dominance. So in incomplete dominance, neither allele has complete dominance, and the phenotype falls somewhere in between the two. So a very simple example is flower color. Again, big R is red, little r is white. What happens if you cross a homozygous recessive with a homozygous dominant? So a big R versus a little r. If you cross them, what do you get? You get one big R, which is red. You get one little r, which is white. But then the two heterozygotes, the big R, little r's, they're pink in the context of incomplete dominance. And that's because neither the red nor the white are completely dominant over the other, and you get these heterozygous pink offspring for flowers. That's incomplete dominance. Next topic you should know is codominance. Codominance is when alleles are both expressed equally. And the primary example that's typically always used is blood type. So let's call the gene I, and you can have alleles A, B, or neither. If you cross a heterozygous blood type A with a heterozygous blood type B, and by that I mean like the AI and a BI, if you cross those two parents, what do the offspring look like? So the type A parent can donate the A allele or the I. The type B parent can donate the B allele or I. And if you now set up a Punnet square, what you'll see is that out of 4 kids, 1 will be heterozygous type A, 1 will be heterozygous type B, 1 will be type O, which is I and I. It doesn't express either the A antigen or the B antigen, while 1 will be type AB, where the red blood cell will express both the A and the B antigens. Okay, and that's what we call codominance. So what is all the fuss about blood type and donating blood? So a type O is the best universal donor because their red blood cells don't elicit an immune reaction because they don't have the A or B antigen on their red blood cells. A type A and B is the most difficult to donate typically because when you give it to a type A person, the body recognizes the B antigen on the red blood cells and recognizes it as foreign and attacks it. While if you give a type AB to a B type person, the body recognizes the A antigen and recognizes it as foreign and then attacks it. So that's why having type AB blood is the most difficult to donate, but having a type O blood is the most convenient or most universal because they don't have those surface antigens. And you can give them to type A, you can give them to type B, or you can give them to type AB, or you can give them to type O. And in all cases, the body doesn't recognize the type A or type B antigen because it doesn't exist there as foreign and doesn't attack it. Okay, let's move on to the next topic, sex-linked genes. For sex-linked genes, the gene of interest is located on the sex chromosome. And we'll just look at red and white eye colors in fruit flies, the typical example typically taught in class. So big R is red and dominant while little r is white and recessive, and the R, like this eye color gene, is on the X chromosome. So out of those 23 pairs, 22 are autosomal and 1 is sex. And so that sex pair on that X chromosome, this is where the gene exists. So what happens if you cross a homozygous dominant red-eyed female with a white eye recessive male? So the F1 generation, all the flies will be red-eyed. This is because the females will get the X big R from mom, and they'll get an X little r from dad, so they'll be heterozygous. The males will get an X big R from mom and then a Y from dad. So all the F1 are red-eyed. The F2 generation is more interesting. Let's think about the males first. They will all, like in the F2 generation, all the males in the F2 generation, they'll get a Y from dad. So the other chromosome will have to come from mom, who's heterozygous. So half will get the big R and be red, while the other half will get the little r and then be white-eyed. So in a way, the mom is determining the trait in the sons because they always get a Y from dad, and then the variability comes from whichever allele the mom donates. How about the females? They'll get a big R, X big R from dad, but half will get a X big R from mom, and the other half will get a X little r from mom. So what that means is all the females will be red, but half will be homozygous dominant, while half will be heterozygous. So that's a simple explanation of sex-linked genes and an example. Okay, let's move on to the next topic, recombination frequency. So recombination frequency is the frequency with which you get a crossover event in between two genes on the same chromosome. So the concept is very simple in that when you have two genes that are very far apart on the same chromosome, the probability is high that you'll have a crossover event in between them, but it gets lower when the genes are really close apart, and that's because there's less space. So if you're going to have like a crossover strike event, if there's this more space in between two genes, it makes more sense that, oh yeah, there's a higher probability of striking between the two. But if they're really close, like you can strike here or here, but not in between the two. So that's the core basic principle, but let's work through a simple example. So say you have genes A, B, and C, and the recombination frequencies between A and B are 20%, between B and C are 10%, and between A and C is 20%. So I'll say that again very quickly. Between A and B is 10%, A and C is 20, and B and C is 10. So what is a linkage map? That's a typical question they might ask in a quiz or a test. And it's simple. It goes A, B, and C. Because the A and B are 10 map units apart, the B and C are 10 map units apart, and then the A and C are the farthest, 20 map units apart. And that's what the recombination frequencies show. So the sequence will go A, B, C, with 10 units between A, B, 10 units between B and C, and then 20 units between A and C, and that's what the recombination frequencies also show. So that's a simple recombination frequency example. Let's move on to the next topic, which is just a concept, and that's polygenic inheritance. So what is that? Polygenic inheritance, all that means is that you have multiple genes contributing to a single phenotype, such as skin color. And in humans, there's something like 378 genes that all contribute to skin color. So that's polygenic inheritance. Polygenes, multiple genes infecting a single phenotype like skin. What's pleiotropy? Pleiotropy is a single gene having multiple phenotypic effects. And for example, if you had a particular allele of a gene in flowers, and that allele resulted in purple petals, but also in the petals being iridescent and shining. That's like two phenotypes, purple and iridescence, and they're both determined by a single gene. And that's an example of pleiotropy. A common example in humans is Marfan syndrome, where you have a single mutation in a gene called FBN1, and the result is that you get multiple phenotypic effects, like increased height and heart deformities and whatnot. And that's an example of pleiotropy. Single gene, a change or an allele results in multiple phenotypes. Okay, and let's finish with pedigrees. So for pedigrees, you just have to remember squares are males and circles are females. You have to come up with some, Nick, some trick in your head to just remember that simple association. But squares are males, circles are females. Let's look at a autosomal dominant trait first, such as widow's peak. So big W is dominant for widow's peak, while little w is recessive for no widow's peak. If you have a heterozygous mom mate with a heterozygous dad, what can you expect in the offspring? Well, out of 4 kids, 3 will have widow's peak, while 1 will not. And that should be irregardless of sex. Okay, so that's a simple autosomal dominant trait. What about an X-linked recessive trait such as hemophilia? This is also pretty common, and common in terms of it being taught in class. So here are some pointers. When it comes to looking at an X-linked recessive trait in a pedigree, I think these are the 3 main things I want you to look out for. Number 1, if a female has the trait, then you know she's homozygous recessive, and that she needs to have 2 of those recessive allele copies to now exhibit that phenotype. So if you see a female that has a trait, then you know her genotype. She's homozygous recessive. Okay. Males will typically exhibit the trait more often, and that's simply because all they need is 1 copy of that recessive allele, and then they exhibit the phenotype. And that's why you'll see it more frequently in males and less frequently in females, because females would need 2 copies. And the last thing you should look out for is that if a female has the trait, all of her sons should have the phenotype. And that's because the sons get a Y from dad, but they always get an X from mom. And if a mom has the trait, that means that she's homozygous recessive, has both alleles that are recessive, and she's going to give a recessive allele to all of her sons, because the sons get a Y from dad and then the X from mom. And mom only has 1 X to give, the recessive allele. And that's why, if you see a affected female, all the males have the phenotype. That's usually a strong indication of an X-linked recessive trait, such as hemophilia. So we'll end there. Those are all the main topics that you need to know for Unit 5 of AP Biology. It was chromosomes, meiosis, where we covered meiosis 1, meiosis 2, the 3-1 Punnet Square, the 1-1 test cross Punnet Square, the 9-3-3-1 dihybrid cross, and then the 9-3-4 epistasis Punnet Square. We talked about incomplete dominance, codominance, incomplete dominance such as flower color, pink flowers, codominance in the context of blood types, sex-linked genes. We did simple, red and white eye color in fruit flies, recombination frequencies, looking at a linkage map, polygenic inheritance, multiple genes affecting one phenotype like skin color, Pleiotropy, a single gene change having multiple phenotypic effects such as Marfan syndrome in humans, and finishing off with pedigrees. So those are all the main concepts of everything that you need to know for Unit 5. I hope you found that helpful, and thank you for listening.